Can food preserved in jars with salted and acidic water lead to the growth of parasites or viruses that cause infections linked to neurological disorders, the brain axis, or cancers?
Preserving food in jars filled with salted and acidic water, such as in pickling or brining, is a common method to inhibit the growth of harmful microorganisms. However, if not done correctly, there is a risk of contamination by parasites, bacteria, or viruses that could lead to infections. Here's how this relates to neuronal diseases and cancers. 1. Parasites and bacterial infections. Improperly preserved food can harbor parasites like Toxoplasma gondii or bacteria like Clostridium botulinum, which causes botulism. These pathogens can cause severe infections that may affect the nervous system. For example, Toxoplasma gondii has been linked to neurological issues in immunocompromised individuals, and botulism can cause paralysis by affecting nerve function. 2. Viruses. While viruses are less commonly associated with food preservation, improper handling of food can introduce viral contaminants. Some viruses, such as hepatitis A or norovirus, can cause systemic infections, though they are not directly linked to neuronal diseases. 3. Neuronal diseases, the brain axis. Certain infections can impact the brain and nervous system. For example, Listeria monocytogenes, which can grow in improperly preserved foods, can cause meningitis or encephalitis. Chronic infections or inflammation caused by pathogens might contribute to neurodegenerative conditions, though this is an area of ongoing research. 4. Cancers. There is no direct evidence that food preserved in salted and acidic water causes cancer. However, certain preservatives or contaminants in improperly prepared foods could potentially increase cancer risk. For example, nitrosamines, which can form in preserved foods under certain conditions, are carcinogenic. Chronic inflammation from infections could theoretically contribute to cancer development over time, but this is not specific to jarred foods. Key considerations for safe preservation. Use proper sterilization techniques for jars and lids. Ensure the correct concentration of salt and acid, e.g., vinegar, to inhibit pathogen growth. Follow tested recipes and guidelines for canning and preserving. Store preserved foods in cool, dark places and check for signs of spoilage before consumption. In summary, while properly preserved foods are generally safe, improper techniques can lead to contamination by pathogens that may cause infections affecting the nervous system or overall health. There is no direct evidence linking such foods to cancers, but improper preservation could introduce carcinogenic compounds or chronic inflammatory conditions. Always follow safe food preservation practices to minimize risks.